Coming up on Quest, a NASA rover looks for signs of ancient life on Mars.
We know it had water, it had rivers, lakes, maybe even an ocean.
A search for clues about a mysterious disease killing millions of starfish. The surprising health benefits from eating dark chocolate. And a mission to uncover the amazing secrets of the spider web. Next on Quest.
Support for Quest is provided by the National Science Foundation, the David B. Gold Foundation, the S.D. Bechtel Jr. Foundation, the Dirk and Charlene Capsonell Foundation, the Vadez Family Foundation, George G. and Jeanette A. Stewart Charitable Trust, and the members of KQED. Quest is a project of KQED Science.
On August 5, 2012, after rocketing through space for more than 8 months, NASA's Curiosity rover began a 7-minute descent onto a crater on Mars. At the time of its landing, the 1-ton SUV-sized rover was more than 150 million miles from Earth, which meant that it took 14 nail-biting minutes for the NASA team at Mission Control to learn the fate of its $2.5 billion rover.
Touchdown confirmed. We're safe on Mars.
The Jet Propulsion Laboratory in Pasadena, California, manages the Mars Science Laboratory mission, which is using Curiosity's suite of sophisticated scientific instruments to reveal tantalizing clues about the existence of ancient life on the red planet. Less than 2 months after arriving, Curiosity's high-definition cameras captured for the first time images of gravel from a stream bed, evidence that fresh water once flowed on Mars billions of years ago. In 2013, Curiosity's analysis of Martian rocks also revealed the presence of oxygen, water, carbon, and sulfides, ingredients necessary to support life. Curiosity has already driven more than 5 miles and continues to beam images and measurements proving that this barren, frigid planet once had an environment where tiny organisms could have lived. But one thing Curiosity can't do is spot current signs of Martian life. Still, Curiosity's amazing discoveries would not have been possible without the advanced engineering behind the scientific instruments that power this high-tech search for extraterrestrial life.
Mars is really interesting for the search for life because it's a world which we know at one time had an Earth-like period. It had water, it had rivers, lakes, maybe even an ocean, and therefore we think it must have had a thick atmosphere.
Christopher McKay from NASA Ames Research Center in Mountain View, California, has spent more than 30 years looking for life on other worlds.
We know that rocks have come from Mars and landed on Earth. We assume that the reverse is also possible, and we know that these rocks could carry life. But I think a deeper and more important question is the possibility that there was life on Mars and it represents a separate origin of life, a second genesis.
Since the 1960s, dozens of increasingly sophisticated robotic spacecraft, including orbiters, landers, and rovers, have been launched from Earth toward Mars. The twin rovers Spirit and Opportunity landed on the surface of Mars in 2004. Natalie Cabrol of the SETI Institute in Mountain View was on the team that decided where to send them.
We went from Mars frozen in time, cold, on the lava plain, to earlier Mars on the Columbia Hills, a time where climate was warmer and geological activity was a lot more diverse. And all of a sudden we discovered evidence of water, evidence for volcanic activity, interacting with water, explosive activity.
And now the Curiosity rover, the largest and most sophisticated science laboratory ever sent to Mars, is collecting more data about the planet than all preceding Mars missions.
You have microscopes and lasers to zap the dust from the surface so that you can look really at the composition of what you're looking at. You will be able to see mineralogy, composition, chemistry. To me, Curiosity is this amazing Swiss Army knife rover of the Mars exploration, you know.
One of the tools in that Swiss Army knife is called CheMin. It was invented by astrogeologist David Blake from NASA Ames.
Yeah, we're in the right place, I think. Yep.
A few years before Curiosity's launch, Blake and his colleague Tori Holer tested a terrestrial prototype of CheMin at Jasper Ridge in the eastern foothills of the Santa Cruz Mountains.
So we're going to do the processing of these rocks just like Curiosity would do it on Mars, but of course we don't have the real fancy hardware that's on Curiosity. We basically have hammers and things like that.
CheMin is able to identify the minerals in samples of powdered rock or soil through X-ray diffraction. Here, an X-ray beam is focused on a sample, and how the X-rays scatter can help reveal the sample's structure and composition.
Just put a little bit inside of that hole. I can see within 10 seconds that this is a serpentine rock.
It takes up to 10 hours for CheMin to do on Mars what Blake's prototype did in just a few minutes at Jasper Ridge. The data from the analysis of each sample goes to the rover's computer, then to an orbiting satellite, and then back to Earth, where Blake and his team analyze the results.
If we find rocks that are similar to what we saw in the field today, we would be able to say that there was water present there, that energy was present, created by those rocks, that could be used by a microbe for life.
As the nuclear-fueled rover transmits its stunning images and carefully collected data, the question remains, could life actually exist on the red planet? Cabral looks for an answer to that question in some of the most extreme and inhospitable environments on Earth.
I have yet to find a place where I did not find life on Earth. In one of these most impossible places, you still find things. That tells you something, that sends you a message. Wherever life can be, it's gonna be.
In 2012, NASA announced Mars 2020, a new mission that will continue to dig for clues of past life and also gather data about habitability of the red planet, information that would be key to the success of future human expeditions to Mars.
I think there will certainly be human exploration of Mars. We'll go to the moon, we'll set up a base there, and then we'll go to Mars. The question is really just when, and will I be around to see it?
Given all the problems we face on Earth, is the search for life on Mars worth all of the effort and expense?
It's just like asking Christopher Columbus, which most of the European kings and queens did at the time, what there is to go and explore. At some point you have to take a leap of faith. But if you don't take that leap, you will never know.
Something strange is happening in the ocean off the West Coast. Laura James was one of the first to notice. She alerted scientists when starfish began washing up on the shores near her Seattle home.
And I thought, you know what, this is getting a little too close for comfort. I need to go see what's going on, and I need to document it.
As a diver and underwater videographer, James was equipped to do something. She decided to take her camera to a spot popular among both divers and starfish. These pilings are usually covered with a rainbow of starfish. On a recent dive, James discovered a scene from a horror film.
There were bodies everywhere, and they were just like these splats. To me, it always looked like someone had taken a laser gun and just zapped them, and they just vaporized.
Starfish, also known as sea stars, are wasting away by the millions, not just in Washington's Puget Sound, but up and down North America's Pacific coast. And nobody knows why.
I've been diving out here for almost 24 years, and people always ask me, do you see any big difference between, now and when you started? And I've seen some subtle differences, but this is the change of my lifetime. We've had small occasional die-offs here and there, but it's not like this. It's not a mass mortality event. I'm just a diver. I need to find out what the scientists know.
But scientists have also been wondering what's going on. They first started noticing sick and dying starfish on Washington's Olympic Peninsula during the summer of 2013. Reports have since surfaced from all along the California coast to as far north as Alaska. And the disease affects almost every species of West Coast starfish. It's been coined sea star wasting syndrome, and it's also been reported at sites along the East Coast, but researchers say it's still too early to connect these outbreaks.
I was surprised too that the crabs weren't just...
Ben Miner is a biology professor at Western Washington University. He studies how environmental changes affect marine life. His team is collecting sea stars at Mukilteo, Washington, just north of Seattle.
The population of sea stars is, they're quite abundant at that site. And so on the pilings, there were healthy sea stars, but we were also coming across arms and piles of deteriorated sea stars and individuals that were twisted.
The divers are searching for stars showing symptoms, as well as ones that appear healthy.
The experiments are infectiousness experiments where we take individuals that have signs of the syndrome and we put them in tanks with individuals that don't have signs.
Then they closely watch the progress of the disease. First, the stars twist their arms into knots, and sometimes lesions form on their skin.
One of them was very sick, and the other two individuals started ripping themselves apart. The arms just crawl away from the particular body.
You heard that right. The arms start crawling in opposite directions until they tear away from the body and their insides spill out. And unlike most starfish, the arms don't regenerate. Stars that came in with symptoms died within 24 hours. Through experiments like these, scientists have determined that the disease spreads through the water and through physical contact. They're also testing the hypothesis that the pathogen may be transferred through shellfish, which starfish like to eat. But scientists still don't know what caused the outbreak in the first place. Could an infectious pathogen from the other side of the world have hitched a ride on ocean-going ships? Or could it be something larger, like climate change or ocean acidification?
So this is a healthy Pycnopodia, no signs of lesions.
Drew Harvell is coordinating the research into answering those questions. She's been studying marine diseases for decades and has never seen anything like this before.
This is the largest disease outbreak that we know of ever in the oceans, in terms of the numbers of species affected, in terms of the geographic scale, and in terms of the mortality that's associated with it.
Harvell is sending these samples to the lab to be analyzed for viruses as well as bacteria and other protozoa. Once the pathogen is identified, scientists will better understand whether West Coast starfish will be able to recover. In the meantime, Harvell is keeping a close eye on the starfish that live near Washington's San Juan Islands. Until recently, the starfish here were spared. Cold water around the island seems to have protected them. But summertime means warmer waters, and stars here are suddenly showing symptoms. Their bodies are deflated, they're unable to hold onto rocks, and their arms are falling off.
I'm expecting that in the next 2 weeks we will lose virtually all the stars at this site.
Today, Harvell is searching for baby starfish. Whether starfish survive on these beaches in years to come depends on these juveniles.
We would really love the sign of hope that maybe they'll pull through, so we're going to be watching them very closely.
If the juveniles can't withstand the disease, it could be a very long time before West Coast starfish recover. And such a loss could have far-reaching ecological consequences.
We're losing a whole guild of stars that are voracious predators, all of which can control their ecosystem. And so to lose all of them at once, we don't even know what's going to happen.
If there's a silver lining, it may be that studying this outbreak could shed light on how marine diseases spread.
2, 3, 4,
and that's a question Laura James is hoping citizen scientists can help answer.
The big problem we had here is that we didn't have a baseline. The starfish got sick when we noticed. Wouldn't it be cool if we could show this in real time and show the spread and the changes in real time?
James and her dive buddy built a website for tracking posts to social media sites with the hashtag #6starfish.
Take a picture of any starfish you find when you're out tide pooling or just walking on the beach and hashtag it #6starfish. And then we can look at it when it pops up on the map, or if we're not sure, we can send it off to the scientists and they can take a look at it. We may not be able to stop it, and we may not be able to fix it, but we need to be aware so that we can recognize it when it happens again.
Typically, most people would just see a spider web and say, ooh, gross, and walk away. When I see a web, I notice the variety of different shapes of webs spun by all the different species of spiders. I'm Todd Blackledge. I'm an associate professor of biology at the University of Akron, and I do research on spider silk and spider webs. I do sometimes get called Spider-Man. Spider-Man, Spider-Man, does whatever a spider can. It's one of the hazards of the profession. Spiders are amazingly fascinating organisms to study. In the United States, I think we're kind of taught as children that we're supposed to be afraid of them, but they're just an incredibly diverse group of animals that play incredibly important roles in our ecosystems as predators of insects. So here's a nice little web in this shrubbery. Out in the field, I can actually collect silk threads directly from spider webs. But in the laboratory, I like to collect the silk straight from the source, from the spider's own spinneret. This is a multi-step process. First, we catch a spider and put it to sleep with a little carbon dioxide gas so that we can gently strap it onto a petri dish using Scotch tape. This holds the spider immobile so we can move it to a microscope and then visualize the silk thread coming out of the spigot and pull that silk thread out. There are spiders that spin up to 8 different kinds of silks, and some of those silks are stronger than steel, ounce for ounce, while others are as stretchy as a rubber band. Some of them are even adhesive, like glues. So this means that they have a toolkit of different types of biological materials that could be used in many different ways for human technology. My job is great. I get to be outdoors in places like this, and I also get to be in the laboratory making new discoveries all the time. It brings something new every day.
Ah, chocolate. It's said that 9 out of 10 people love it, and the 10th person is lying. Chocolate's universal appeal dates back 2,000 years to the ancient Maya and Aztec civilizations. These cultures believed that cacao, the tropical plant that produces chocolate, was imbued with divine powers. Cacao seeds were used as currency and to improve health. And today, scientists agree. Chocolate is actually good for you.
The exciting news with our study is we found that small daily doses of dark chocolate could actually improve vascular function.
Mary Engler has studied chocolate since 2001, when she was a professor at the University of California, San Francisco.
Dark chocolate is within the one category of flavonoids called flavonols. Within that group are a number of compounds that have catechin or epicatechin.
Catechin and epicatechin are naturally occurring compounds that increase the levels of nitric oxide in your bloodstream, causing your arteries to dilate. This allows your blood to flow more freely.
We know that heart disease, you have narrowing of the arteries and blood flow can't get through. Blood flow is very important to help prevent any kind of cardiac event.
But all chocolate is not created equal when it comes to benefiting your heart.
Dark chocolate is much better because of the high content of the cocoa flavanols.
The flavanol content of dark chocolate is even higher than that of other flavanol-rich foods like cherries, red wine, and tea. It not only supports circulation, it also soothes frazzled nerves and lightens gloomy moods.
You can raise your serotonin levels, and that's why you get the good feeling after having dark chocolate.
Chocolate may be especially helpful for pregnant women. A 2004 University of Helsinki study showed that women who eat it every day during pregnancy report calmer, happier 6-month-old babies. It also reduces the risk of preeclampsia, a dangerous pregnancy complication. But perfecting the taste of chocolate is trickier than you might expect.
So that's what the final chocolate looks like.
Meet Timothy Childs, one of the founders of Cho, a pioneering artisanal chocolate maker based in Berkeley, California.
Chocolate's an incredibly complex system. It goes beyond just hard science. You also have to integrate art and a lot of intuition, too, at the same time. So it's a really interesting combo.
Any fine chocolate begins with the cacao tree. A mature tree produces about 20 fruits or pods, each containing about 40 beans. An average tree produces about 2 pounds of chocolate per year. After the beans are extracted from the pods, they need to be fermented. Fermentation halts the seed's growth and develops the cacao's complex flavors.
The traditional style of fermentation is in big piles, and that doesn't have proper kind of even cooking of the beans, and so we try to encourage our farmers to go to a more of a box method.
After fermenting, the beans need to be dried. Traditionally, growers dried their beans on concrete, but Cho found a better solution.
When the beans are drying, they're outgassing what we call volatiles, which are pretty much acidic acids and other undesirable flavors. But if you're outgassing on plastic or concrete, it bounces right back into the bean. So if you actually dry on bamboo racks and stuff, or anything that's got air moving underneath it, those acidic acids combine with the oxygen and blow away.
To maintain quality control, Childs created an innovative device to connect with his growers in Peru.
We pretty much took an off-the-shelf weather system and did some custom modifications to it. This weather system sends weather data wirelessly to a laptop, and that laptop's hooked up to a cellular modem that sends it up to the internet.
Cho employees use this data to track how the weather affects each batch of beans. Then, when the company receives a superior batch, there's no mystery about the conditions that produced it. Cho also created a series of flavor test labs in order to produce tiny batches of factory-grade chocolate right where the cacao is grown. The labs allow farmers to taste their own product and to bring entire harvests up to a shared standard for taste and texture.
Look at the difference in the texture. See how it's got really good definition? This tastes good, this does not taste good.
Here in the company's original flavor lab, the raw beans are first roasted in a modified turkey oven. The beans are then cracked open, and another homemade contraption is used to separate the nibs, or the meat of the bean, from the shell. Next, the nibs are ground with a repurposed Indian curry dal maker. The refining stage determines the chocolate's texture and creates the smooth mouthfeel that aficionados savor. Then the chocolate is conched or heated and cooled repeatedly to fine-tune its most subtle flavors. For Childs, the real magic happens during the tempering stage.
You temper chocolate just like making tempered glass or tempered steel. You start with a molten fluid, you seed it with a piece of already crystallized material. You're actually creating this molten lattice crystal structure that's flowing. And then if you mold it and you cool it just right, what ends up happening is all these little fat molecules kind of line up, and then they get tighter and tighter and tighter until it goes and it fuses in this crystal. And that's what gives chocolate its snap.
For the 6.5 million farmers who grow cacao, the crop is as precarious as it is precious. Every year, nearly a third of the cacao crop in Africa is lost to pests, drought, and disease. Losses to farmers around the globe amount to $800 million each year. In 2010, the genome of the most popular variety, Theobroma cacao, was sequenced by scientists working with the USDA, IBM, and Mars Chocolate. To decipher the cacao DNA, scientists first crushed the leaves and pods, then extracted the nucleotides that make up the plant's genes using a DNA sequencer. With free online access to the sequenced cacao genome, farmers should be able to identify the tastiest and most productive plants to breed. This could help ensure the sustainability of cacao farms worldwide, with crops that can withstand assaults from disease and a changing climate. And that's sweet news for the future of one of the world's favorite treats.
Support for Quest is provided by the National Science Foundation, the David B. Gold Foundation, the S.D. Bechtel Jr. Foundation, the Dirk and Charlene Capsonell Foundation, the Vadez Family Foundation, George G. and Jeanette A. Stewart Charitable Trust, and the members of KQED. Quest is a project of KQED Science.